So let me give you a little bit of background about the presentation and how I've modified it. This is the second time I'm giving this talk, so it's kind of early days for the topic, or at least the presentation. So when I moved to Penn State, on my floor, I had the privilege of sharing space or an area with Masato Shine, who is one of the most important evolutionary geneticists of the 20th century, and I was very intimidated, and he didn't know that I had some background in evolution. As Bill said, I've been doing more sort of mechanistic, physiological, functional genomic studies in the last few years when I got into coral research. But my background is in comparative biology, and in particular, Metazoan biology. I'm an invertebrate zoologist by training. So he came up to me and said, he used to have an institute, he has now retired. He said, I want you to give a talk for the institute. And I was, how do you say no today? You know, and I said, how would I say no to you? And he said, yeah, you can't say no to me. So I had to put a very conceptual talk, and I said, we're not doing much evolution, but we have a little bit of this. And it got me thinking sort of dust and overcome the fear of putting out some of these ideas that are kind of somewhat philosophical, and there is never a right answer to this, and use what we've been doing in the lab to try to address sort of theoretical evolutionary questions. But I've thrown in enough, I think, stuff that may interest the coral crowd and other people. So I'll just mention those studies in passing, in case you want to talk about that later on. I'll be here today, tomorrow, and then the rest of the weekend. And I'll be giving a more general presentation in the lab meeting with Bill and Tracy and Betty tomorrow. If you want to hear about the bleaching and the other developments, symbiosis-driven development and microbiome stuff. That is more casual, just to open the discussion. So if you're interested in those topics, you have another opportunity to discuss them with me. Okay, so the initial talk was about insight from coral, but then I learned that Dave and Aurelie are working in mollusks, so I included that in there. So these are the study organisms, not this coral in particular, but corals. A land snail, freshwater snail, Biominerals, Brata, and Aplysia, and Opistobranch, and a few other organisms that you'll see throughout the talk. So enough of an introduction. What's happening? There. So this is what I was referring to. I've been thinking about, I did my PhD in Opistobranch evolution, which is a place in the gastropatry where there's a lot of convergent evolution. So there's been evolution of shell laws multiple times in these organisms, and they really got me thinking about the difference between homologous traits and traits that are not, which is what homoplasy is. So don't think too much about homoplasy. Can be a very controversial topic, but the question that I'm after today, or try to get across, is trying to understand homology and why this is important. So even though it's been since Darwin times, and there was debates between Owen and Darwin, and Owen was not an evolutionary biologist at the British Museum, and Darwin was pushing evolution. The concepts were defined by museum people that were not really thinking in comparative terms, and I think that's where the philosophical conflicts start. But we do know that certain things are the same. Oops. So limbs, invertebrates, even though they have different functions, we know they are homologous traits. Wings in insects and vertebrates are not the same. So this is a case of homoplasy, this is a case of homology. So shells, skeletons, vertebrae in animals, are these homologous characters or not? A lot of people would argue that they aren't, and some people would argue that they are. So this is the system that we chose with one of my grad students to try to contribute to this debate, intellectual debate. Why? It's a very important question in terms of Metazoan evolution, because for 540 million years ago or so, during the Cambrian Radiation, you see this major appearance of all animal phyla, most of them calcified in one form or another. So are these things all sharing an underlying mechanism that drives the formation of their skeletal structures? And this happened in a very sudden period, so 10 to 20 million years, and some claim, I think this is a little bit anthropocentric, but some claim, at least it's been widely studied, the most dramatic adaptive radiation in the history of life. Certainly the most widely documented and really fascinating from a paleontological perspective. So this is where I'm trying to squeeze some of the things that are relevant to what coral reef biologists think about today. Ocean chemistry, which is depicted here at the bottom, varies over time, has varied. So here in blue you have what they call an aragonite sea, so chemistry that is conducive to aragonite skeletons. And then later on, mid-Cambrian and on, through the Ordovician, you have a calcite sea. So if you look at the types of skeletons that all these invertebrates are forming and previous ancestral forms, you have aragonite type calcium carbonate early on in the Cambrian, and later you have more of a calcite carbonate. So ocean chemistry and the type of skeletons and organisms that live in these oceans is complementary. One drives the other. At least that's the hypothesis. This is a work by Suzanne Porter, who came out of Andy Knoll's lab at Harvard. She's a professor at UC Santa Barbara now. And this is when Bishoy, an extremely skilled, soon to graduate and in the job market, PhD student, joined my lab. And he said, this coral stuff, it's not for me. I don't like diving and swimming, and I want the computer stuff, so I want to work on the Snell project, although he's been co-opted, as you'll see later on in the talk. We decided to pursue this, and this is why. So this is a quote from this review by Andy Knoll, a paleontologist at Harvard. The many origins of calcareous skeletons reflect multiple independent cooptations of molecular and physiological processes that are widely shared among eukaryotes. So not just metazoans, but eukaryotes in general. Skeletons that are not homologous in structure, so that snail shell and that snake vertebra are these structures, share underlying physiological pathways that are. And that is what we're trying to prove in this dissertation. This powerful idea has to be correct at some level. We believe it is. The details, how much biochemical innovation and diversity underlies carbonate skeletonization across clades awaits, sorry for the typo here, oops, an expanded program of comparative biological research. And that's when Bisha said that's going to be my dissertation 5-6 years ago, and you'll hear the beginning of that. So when we started to go to some meetings and talking about this, it was surprising that people were like, what is biomineralization? So even though I don't think I need to define that to this crowd, I will. It's a highly controlled and functional process of hard tissue formation. So this is important, highly controlled and functional. The ability to calcify a supporting skeleton was a major evolutionary transition that contributed to the Cambrian radiation. Why? Because a biomineral without proteins is just rock. So this is a biomineral, this is a rock, this is a quote from Weiner and Addy, to the leaders in the field in biomineralization, Israeli scientists at the Weizmann Institute. Okay, so if you map calcifying organisms onto a phylogeny, this is a representation of the types of molecules that you see, calcium carbonate being the most predominant one. So that's the one we've chosen to focus on. But there is some other types of biominerals, like siliceous-based. So biomineralization is an emergent property. It's complex, and there is many challenges to define what it is and what is homologous and not. Why? Because there are multiple processes that are required to obtain a final product. So you need an extracellular matrix that organizes and drives the formation of the biomineral. You have to regulate how the mineral is being precipitated. You have to inhibit growth, and that's something that is actually very important in biomedical research because there is a lot of bone deformities that are caused by failures in the inhibition process. And then you have to transport calcium across membranes. So there's probably other aspects of the process that we don't even know about that also are important in trying to understand these emergent property. So Knoll proposes three hypotheses about the evolution of biomineralization. One, that it independently evolved over 10 to 25 million years in 35 animal phyla, which seems highly unlikely, in my opinion, or that it is equally unlikely that it is an unprecedented event of lateral gene transfer across the different organisms in the Cambrian, even though there is more and more evidence about lateral gene transfer in eukaryotes. I don't think that this is as predominant a process as it is in prokaryotic organisms. So this is the one we favor, natural selection through geologic time from an ancestral biomineralization toolkit to undetectable homology today and at the level of an anatomical structure, so a shell and a coral skeleton, a gastropod shell and a coral skeleton. So we started with a computational approach. What was there in the literature? So we gathered all biomineralization proteins from different phyla, and when I say all, it sounds like a lot. It isn't, and you will see in a second. We broke each protein into domains, protein family domains using hidden mark models, and then use those protein domains to use interactome data, protein-protein interaction data, PPIs, from interaction databases from model organisms to increase, sorry, this is a new pointer, I'm still learning how to use it, to increase the pool, the starting point of biomineralization proteins in any organism, and then compare these across animal lineages. So, I don't know that for the sake of time, I'll go through these very quickly, but if you want to know more, I can tell you more about this. This is already submitted anyways. So, literature. We went to the literature is extremely biased. Pearl, biomineralization in bivalves, vertebrates, bone disease, tooth formation and then sea urchin development, primarily, very, very few. So with that, we started a candidate list, and then we performed a domain PFAM search using the hidden Markov models. We came up with 198 protein families that seem to be involved or have protein domains that appear to be involved in biomineralization processes based on the literature. And then we also use BLAST to identify in the databases and EST genomes and ESTs publicly available at the time to come up with new ones. And then there was a series of checks to corroborate that this was a good prediction. The cutoffs for the BLAST searches were really, really, really stringent.
So you're including in the protein list for consideration transcription factors, signaling molecules, not just structural proteins, right?
Anything that we found in the literature associated to biomineralization. And this is the initial number, 474 proteins. This is very old when we started doing this. And this has, Bisho is going through multiple iterations of this. But this was the starting data set at the beginning of his dissertation, 72 proteins from molluscan literature, 55 from the urchin spine development, Evo-Devo studies, only 15 from corals, and then from Amigo geontology. And then those that were 100% identical were removed. So this was the initial starting set. And then we partitioned this and were looking which ones were conserved across these lineages that were the ones that came from the database. So it's a little bit tautological, it's redundant. We're going full circle here. But we identified 81 that were present in all the lineages that were available when we started. The problem is that these public databases are biased because of the type of research that people are doing in terms of biomineralization that are taxon dependent. There were not many other mollusks, and which is what my lab was working on, and certainly not any basal metazoans. And I won't tell you about the brachiopod story because it kind of failed. But we were trying to get some brachiopod stuff. So the solutions were to organize the information. So Bishoy wanted to construct this database and then generate new data. So we sequenced a bunch of transcriptomes and took advantage of the genomes that have common line for these organisms, and we chose the organisms based on what was going to come online for the molluscan genomes, and then we sequenced a coral genome, and I'll tell you very briefly about that, but we can talk more about what we're doing with that. And then test hypotheses about the function of homology of these, and I won't spend a whole lot of time on that, but I'll mention it briefly. So BioMine is in review. Basically, is the information that we gathered from the literature, and using the pipeline that I explained to you, you can search by species and you can search by protein, and then this is sort of an output of what you get, and you get useful information like the type of, you get the sequence, of course, the isoelectric point of sequence, which is important for biomineralization studies. And then if there is a publication associated with it, you'll find that. So I'll come back to that in a second, how we expanded, and we will continue to expand BioMine. But I'll tell you now about the transcriptome studies briefly. So because there are database biases, we wanted to include something basal at the base of the tree. There was nothing. Now there is the Acropora, Millepora, Proteome, and it's Xylophora stuff from Pakowski's lab.
Sponges too.
Yeah, right. Sponges. So there it's just exploding. But when we were thinking about this, there was really a database bias. Mollusks, even though they all form shells, or the vast majority of them form shells, there was very little information. So, because we know which is the tissue that secretes the shell, and it's somewhat accessible, easily accessible relative to corals at least, we went for a mantle tissue transcriptome, so looking at the genes expressed in the tissue that make the shell. We chose 3 gastropods and 1 bivalve, and again, this was driven by what we knew was going to be publicly available in terms of genome reference information, and also phylogenetically, strategically placed Biominerals. So pulmonate snails and Opisthobranchs are sister taxa, so the annotation is more feasible, or easier to handle. And these guys, the seahairs in the adult form, don't have a shell. And this is a question that I've always started tinkered with. We are finding some interesting stuff there. And then the Pacific oyster and Lotia, which had the genome available. Did RNA-seq, and then the genome projects came online with their own tissue-specific transcriptomes for gene prediction. So those data sets are really, really high quality, and that's what we ended up using. So we did the same sort of analysis, and this is what you see here. So we again identify a significant number of core proteins that are shared across these 2 classes of mollusks, at least, bivalves and gastropods. And we also find some other interesting things. So things that are unique to those that make a shell. So Aplysia doesn't make a shell, adult shell, and it's a chitinous shell. It's not a calcareous shell anyway, it's the remnant. things that are unique to those that live in seawater, and because Biominerals is the vector for schistosomiasis. It's a freshwater snail, so the shell structure is different. So this was a very useful kind of iterative process that we could use to analyze the mantle transcriptomes. And there is a little bit more bioinformatics in that because we use the genomes, but anyway, we were able to kind of keep using the same strategy in an effective fashion. So this brings us to this question of homology. You have all these interaction networks, protein-protein interaction networks that act at different levels of organismal specificity. So you have things that are very universal, things like actin that everybody has in every cell, that are conserved, and then you have sort of middle of the pack proteins that you may find across certain lineages, but then you have taxonomically restricted proteins that are specific to make a certain structure, either the skeleton in a coral or the shell in a snail, like osteocrine or chimatrines or other taxonomically restricted proteins. So all these proteins, however, interact with other proteins. And that is, I think, where BioMine becomes really helpful. You go into the interactome data sets and you can then pull from those interactomes new candidates. And this is something that can be used not just for biomineralization, but for other types of questions, biological questions, where you have some candidate and then you go in and tap onto the PPI databases. So you identify things that are highly conserved. That's what these hubs represent in these protein-protein interaction networks. So these are things that we know about. We've known from cell biology studies. But then you know the things that have unique links to that hub that they may be new proteins that have only been revealed in your transcriptome that now you know interact with that particular protein that has a biomineralization protein domain. So that's how you can increase your list of candidates, and you'll have potential interactome hypothetical data that then you can pursue and use. Basically, reduce your search space significantly that way. And if you have some other sort of evidence that suggests that your protein is involved in these particular processes, then you can go into BioMine and corroborate, and then you'll have bioinformatics support independently for your candidate protein. Okay. So yeah, so you have conserved things, and then you have nodes that are not such great hubs in this network, but they interact still significantly. So those would be those sort of middle of the range in terms of organismal specificity in the protein-protein interaction network. So the core in the middle forms more or less a solid unit. This network contains new PFAMs. So the ones that we started with, 198 candidate PFAMs, now it's expanded by the interactome map. And as I explained, those are the new candidates then. So going back to this framework, when we looked at these proteins and tried to map them onto this diagram, we found this is how you can visualize the interaction. So a lot of them are involved in regulation. There is some of them of the known proteins that are involved in extracellular matrix and transport, but we didn't really find many things involved in inhibition, and that may be a database bias, or also because those processes are very unique to each organism in terms of how the calcifying structure is being formed, is downstream in the process. So that's sort of the working hypothesis for that. Okay, so the Orbicella genome. So we work on this organism extensively. That's what has been the thick of the work, actually, and that's in my lab for the last 10 years or so. And that's why I sort of didn't want to tell you about that, because you have probably read it, and some of the stuff that is ongoing is just too new. But we were pursuing the genome for many, many different reasons, and then Bishoy got involved, and now he's a coral reef genomicist. So one of the things that is difficult with coral is that the calicoblast ectoderm is difficult to isolate. So trying to do a transcriptome study involved more sophisticated techniques that we didn't want to pursue. We, in collaboration with Frederick Moran, have pursued the proteome. I'll just tell you a little bit about that. But we figure we're sequencing the genome, let's just tap into this bioinformatic resource and try to advance knowledge that way. So this genome is pretty large, 100 megabases, so it's not as small as the published Acropora digitifera genome. But we got a pretty good assembly now, and very, very good gene models, gene prediction. It's a really nice basal Metazoan genome. Okay. So what do I want to tell you about this? So this is a summary of what is sort of in the literature. 34 different proteins are known from the extracellular matrix, in addition to other transported proteins from the published literature, Acropora and Xylophora, and we found 29 of which are in Orbicella, in our genome. Of these proteins known are really strictly unique to corals. 17 of those, the carbs, are found in mollusks and in other metazoans. So maybe these are some of those core conserved proteins in that Cambrian pre-Metazoan biomineralization toolkit. And a lot of these are, so zooming into the carbs, these are acidic proteins, and what has emerged from using the genome data and transcriptome data is that these genes, these biomineralization genes, are encoded within other proteins in many, many instances, and I'll show you an example of that with this CARP4. And the gene structure is conserved across species. However, there is single point mutations that we encounter, and these can change the isoelectric point, which might have a strong effect in calcium binding and potentially impact the biomineralization process. This didn't translate very well, but still you can see it. So I'll start at the bottom here. So you have two transcripts. This is a neuronal acetylcholine receptor. Okay, here. And then you have, oh no, the CARP is your query, sorry, and you have the neuronal acetyl receptor gene. So they overlap in location, chromosomal location. So if you look at the scaffold where you find it, this is the size of the scaffold, 138 kb, and you map both all the reads, transcripts onto the scaffold, you see that basically they overlap to the same region. And
sorry, the genes overlap, or they're located close together?
No, they completely overlap.
So they're separate transcripts?
Yeah, they are separate transcripts. Right. And I just learned about this last night.
The calcification protein is evolved from something that was
yes, and that is what I think I find really interesting, and maybe on something here of why it's so difficult, because there is something shading that, but also at the same time protecting that from environmental. I'll get back to that, and hopefully I'll make myself clear. So I'm sorry it's rough around the edges, but this is just very, very new. I don't know what this is exactly, but yeah, No, I don't know what these numbers are, actually. I'm sorry. But we have the Orbicella is a species complex. So 3 closely related taxa, they're 500 million years apart. No, 500,000 years apart and 1.5 million years apart in case of the deepest divergence. And we have the 3. That may be the isoelectric point that Bishru was able to predict. I forgot to ask him about that. But so you have here 3 of these proteins, and we have them in the 3 genomes, and now we know they're encoded with nucleotide variation. That's probably what this is, the point that I was making in the previous slide. That's a SNP, a single nucleotide polymorphism, can really change the isoelectric point of that protein, and that can have subsequent effect in biomineralization. Which is cool. If you think of small skeletal differences within closely related species, right? It's fun. Yeah, it's a fun hypothesis. So yeah, I think this slide is to illustrate what we can do with the genome to answer very many different questions, right? And a very exciting result. So it seems, from what has been communicated to me very recently, that many of these proteins are encoded within other conserved genes. So, does this support the evolutionary resilience of a core biomineralization toolkit? And I would like to brainstorm with you and with you guys, biomineralization people, about this. Maybe because this proteome exists as part of another proteome that has conserved functions, then things like change in ocean chemistry don't affect this core proteome to the extent that they could, pH changes and things like that, because there is such a strong selective constraint in some conserved genes that are hiding, you disagree, which is good.
So, I mean, it's really cool, it's very, very nice, but I think what you're seeing is evidence for, recent recruitment.
Yeah.
So, my view about calcification and its evolutionary conservation is the basic nuts and bolts, carbonic anhydrases and so on. All of that is conserved, but the control of the skeleton formation, that's taxon specific.
Yeah.
And what I would say, you've got evidence there that these control of biomineralization proteins have evolved recently from...
Like the inhibition and regulatory proteins.
Yeah, so you've got evidence for birth of a new gene family. I bet you that's a new, very recent thing.
It's possible, yeah. I mean, this is a very, very recent thought. So that's why, it's an equally plausible hypothesis. And we can talk more about that at the end. So anyway, the result, I think, is really, really interesting. And it can set the stage for many interesting hypotheses. Okay, so I think I'm running out of time, so I'll just breeze through the rest of this. So, there is always questions about in silico studies, and Bisho is very good both at the bench and at the computer, which is a mixed blessing because he's not so good at writing, and he has so many things to publish. But I'll show you some of the things he's done in case you want to know more. So he went in and tested, I don't know how many in Biominerals. These results here are from Biominerals, and this is from Ilianasa. Another thing, a ton of work. So he grabbed his list and he said, I want to make sure that these things that I found in silico, from in silico predictions, are expressed only in the mantle. So he went and did all these RT-PCRs for close to 100 genes to show that they are indeed mantle transcriptome encoded proteins or expressed proteins, I should say. And then he took some of those in collaboration with the people doing the Biominerals genome and tried RNAi, and he likes to joke that he managed to make a dent in the shell, which is what you see here with RNAi. And then he did a bunch of in situs with a completely different system. I don't know if any of these things will see the light of day because he moves on to other things quickly, but if you want to talk about that, we can. And then we went to Frederick Moran's lab and did a bunch of stuff with the Orbicella proteome. And one thing that I like to highlight, because I think really interests this crowd, is that we've been using corals that are propagated at the Aplysia hatchery in Miami. They have a coral hatchery there too. So these are very clean skeletons, but they're different ages too. They start with really small nubbings and then they grow. So we took a bunch of those. And then Bishoy ran out and he called and said, I need some ground skeleton. And the only person that responded was a student studying disease, yellow band disease. So he sent that. And every test they did gave a very different result. So this is, I don't remember what kind of tests they did. So they did all kinds of biochemical tests with ELISAs and things with Frederick. But the yellow band, sample really stood out as being very, very different in terms of the extracellular matrix composition. So I think that's an interesting result that would be worth following up. And then Bishoy looked at the gene expression data we had, and then there is an increase in gene expression on that particular protein, which is one of those SOMPs that I've been talking about. So it's upregulated in the disease condition. So to conclude, I think the jury's still out, as you saw with this brief exchange that I just had with Dave and about these concepts. But it's something that has inspired many bright evolutionary biologists. There has been a series of books, influential books. This one by Brian Hall in '94. Then there was one sort of the counter of that, presented more by the phylogeneticists. This Brian Hall is a developmental biologist, thinker in terms of comparative developmental biology, and these guys are more phylogenetically oriented, so tree thinkers. And then there was sort of another response to that, compiled again by Brian Hall in '99. And these are some excerpts from that, so that you see where sort of things have gone. Homology is the hierarchical basis of comparative biology, undoubtedly. It can be studied at all levels of biological organization, from molecules through genes, cells, organs, embryos, organisms, populations, even ecosystems perhaps. And this is a quote from Brian Hall. But if you go back to like the 1950s, we haven't really come very far until recently. In many respects, homology remains in '94 where it was back then. After examining the present status of the concept of homology, one arrives at disquieting results. A basic term of one of the most important zoological sciences, that of comparative anatomy, cannot be exactly defined. But I'd like to conclude on a high note by David Wake, a professor at Berkeley that has thought about homoplasy and homology for a long, long time. The integration of genetics, signaling patterns, and regulation of developmental pathways and phylogenetics is in its infancy, but promises to open the black box of phenotypic evolution. So hopefully, we're getting closer to be able to crack this question in terms of whether these anatomical structures are homologous at some level or not. And with that, I'd like to acknowledge this incredible work that has been driven by Bishoy. He took it, it's reshaped my thinking of the capabilities of a PhD student and independence. He's really, really a brilliant student. He recruited a very good undergrad to do all those RT-PCRs, PanQuo. And then these are all the collaborators that contributed either samples or early access to their genomes and training, and then the funding sources, which has been primarily an unfunded project, I should say, but Bishoy has been successful at getting small pots of money from developmental biology societies for students. And with that, I'll be happy to take questions. How did I know that? Okay. Yeah. Thanks, Monica.